Hello everybody, this is Group 4. I'm Uzair and together with Fulya, Tuğrul and Ömer we'll be covering the second half of the article on deep learning with microfluidics for biotechnology. These are the sections that we'll be covering in today's presentation. So I will start with image to unstructured neural networks. These are deep networks to handle spatially distributed data or big set of images. And these are really helpful, especially in microfluidics for classifying cells directly without having any prior extraction of traits. It saves a lot of time. There is no human bias involved in this because everything from start till end is done on computer and hence all the results are much accelerated. So let's have a recap of what neural networks are. Let's write a tree in a 28 by 28 pixel grid. So our brain is able to distinguish it specifically as a tree, and it can do so if we write this tree in a different fashion. So our brain can tell all these numbers to be 3, despite that the specific parts of the brain, the light sensitive parts for each number that are activated are different. So there is some phenomenon happening in the visual cortex of the brain that allows us to differentiate between different numbers and to tell the same number if it's written in a different fashion. For computers, we want to have some similar approach whereby a computer doesn't compare each and every pixel, but it can tell that this is one certain number regardless of the way it is written. So for instance, if we take again the same 28 by 28 grid, this time we write a different number. We assign each pixel with a grayscale value of the color that is given in that pixel, because computers, they see everything as numbers. We call this number as the activation number for a particular pixel. And since we have a grid of 28 by 28, we have a total of 784 pixels, and we need to operate on these 784 pixels, do some manipulations in these intermediate layers, and have an output layer where we can tell that what number this is, again by having a value in the output neuron. These are essentially, when I say neurons, these are just things that hold a number between 0 and 1. And all these lines that you see between different layers, they are some sort of weighted manipulations from one layer to the other layer. So in the traditional neural networks, each layer sends its data to each cell in the subsequent layer, and obviously this would give a lot of calculations. It would require a lot of calculations, and hence it would be a relatively slow process. Previously our image was in grayscale, but if our image is colored, we'll have 3 layers, and essentially we'll have to multiply our grid by 3. So we have a grid of a bigger grid of 200 by 200. We'll have to multiply it by 3, and if we multiply these 3 numbers, we get 120,000 just for one layer, and hence using traditional neural networks requires a lot of calculations. We need a way to reduce the number of calculations. This is where convolutional neural networks come in. The idea is for each neuron to be connected to fewer neurons in the previous layer, and hence reducing the number of computations and the number of neurons required. And this idea is actually inspired by the visual cortex of human brains, and what happens is that different parts of the brain are activated by different signals. Some are activated by edges, some are activated by curves, some are activated by diagonal edges. How CNN works, we are going to use example of 2 characters which we can show can be written in many ways. For instance, the X can be written in these 4 ways shown in the picture. Using normal techniques, computer compare two images by bit comparing their data. And if we do this for the example that we previously mentioned, and we take 2 of the images for X and we compare them, we see that many of the blocks on the right, they don't match. So traditional approach cannot be used to identify any character. Do in CNN is, as I mentioned earlier, rather than looking at the whole image and comparing it at once, we scan through the image and look for similarities in sections. So as we can see, we are comparing different sections on the image on the left to different sections on the image on the right. And when we see this similarity, we can tell that these two are essentially the same alphabet. This approach of CNN can be used in different microfluidic applications, for instance, to calculate the binary population of different types of cells, for instance, lymphocytes and red blood cells at high throughput. This was done by Yo and his colleagues and can be seen in the image below as well. Can also be used to predict different flow types for different types of channels, as can be shown in the image. And they've also been used to quantify bacterial growth in microfluidic culture systems.
Hello, now I talk about image-to-image neural networks. These kind of systems are used to predict a next frame in video, or predicting or evaluating the depth of an image, depth in an image, and also for segmenting images, like differentiating the objects in the images. How it's done is there are 2 approaches to this image-to-image neural networks method. One is the encoder-decoder mode. The other is this spatial pyramidal pooling approach. In the first one, encoder-decoder one, you get images as shown here. The images to the left, 2 colored images, are SEM images of nerve cells. And these cells are segmented throughout this process. The image of cells actually segmented. How it's done? They have this input image, as you see in black and white. And this input image is segmented or separated into layers, as shown in the deep learning architecture. It's shown in green and purple here. And once they separate these layers, they reconstruct these images by using deep learning. In this one, it's called UNet model. And once they successfully do it, they segment the cells in image like this, like the image on the image to the right, the last image, which is called output. As you see here, the blue parts are the axon, the inside of cells, and red parts are the myelin, which are like borders or the membrane of the cell, and black region is the background. And another approach to this alternative approach to image-to-image neural networks is spatial pyramidal pooling, as I said before. In this one, several filters at different resolution layers are applied onto image, and the image is processed this way. And this method separates the task of downscaling and convolution. In the before one, you see the downscaling and convolution was made together, but this one separates it. So this gives us better results when segmenting multiple image scales than the other one. Now the ultimate method is hybrid approach, which is used in video processings. By hybrid, it means it's combination of before mentioned methods. An example in the literature is that CNN and RNN are used to predict single-cell lineage. How they did it was they took local image features out of stem cells by extracting bright field CNN. After this, they get bright field vectors of the cells. Then these vectors are fed into RNN process, and this temporal information that is collected is used to analyze the next frame, possible next frame in the video. And using this method, they image processed almost like 6,000 cells in over 400 sequential frames. And this prediction was successful for observing differentiation of cells even in 3 generations. As I said, the hybrid is the ultimate of these methods, combination of them. Now, Fulya will talk about emerging technologies.
In this part of our presentation, I will provide some information about the opportunities that arose from the connection of microfluidics and deep learning analytics. I will talk about these emerging opportunities in 2 different contexts. The first one is organ-on-a-chip and AI autonomous living systems, and the second is deep learning powered experiment design and control. Organ-on-chip systems are engineered devices that rely on the integration of advanced microfabrication, microfluidics, living cells, and biomaterials to create human tissue constructs that accurately mimic the structure, fashion, and microenvironment of real tissues. Organ-on-chip systems have already been developed for many organs of the body and have demonstrated very high potential for drug development and personalized medicine. That we expect organ-on-chip systems will mimic real tissue with increasing accuracy and complexity that any deep learning application for real tissues can also be applicable for organ-on-chip systems. First, as organ-on-chip systems continue to evolve, microfluidic tissue level structures will be cultured, maintained, and could be studied over large sample regions of the organ-on-chip tissue to detect spatial heterogeneity similar to modern histopathology analysis. And this allows us to generate or obtain enormous datasets of images and videos of living cells, tissues, and organs in their respective in vitro microenvironment and undergoing changes in behavior and function. This data will provide a rich dataset for deep learning architectures. Secondly, considering the emerging vision of the body-on-chip for clinical applications and personalized medicine, there is a potential role for deep networks and AI in the control of the entire multi-organ system. We can imagine deep networks monitoring individual organs, metering communication, providing real-time control of multiple organ-on-chip systems, and working towards a fully trained and AI-guided multi-organ physiological system that regulates itself. A learning AI-driven multi-organ system may in fact be more fundamentally natural, self-guided than human-controlled organ microsystems. Thus, we envision a clear and immediate role for deep learning in accelerating data analysis from organ-on-chip systems and long-term integrated control of multiple organs or tissues. While the majority of near-term deep learning applications will focus on post-experiment data analysis, there is also a growing potential for designing microfluidic systems and controlling systems during experiments. For example, there is a synthetic biology company which controls thousands of parallel microvalve-based microbial culture experiments where the fluidic decisions are made autonomously via AI. In this case, deep learning analytics and fluid experimental setup work together. And the second example, neural networks have been applied to predict the outcome of organic chemistry reactions, and also deep learning analytics can be applied into fields with deeply complex and convoluted factors, such as the research of such as research about effect of environmental stressors on microbiota in the awareness of climate change and pollution. That in order to quantify the response of complex systems to multiple stressors, this will require high numbers of parallel experiments in both experimental planning and post-experimental analysis.
Now I will talk about globally distributed microfluidics with cloud-based deep learning. It means that the microfluidic systems can integrated with machine learning to improve their abilities. As we all know, paper-based microfluidic assays today provide very rapid results in terms of giving or separating cells or other biological materials or other environmental materials. However, the imaging systems slows this rapidness. And integration of these with smartphones that we all use today would increase this develop this technology a lot, and this would allow a much better deep learning mechanism globally. For instance, for point-of-care diagnostics, globally distributed users can generate huge amount of data from all around the world. We could get point of diagnostic results into our system, and we can track this data or this data with this system, so that we can track or sometimes predict diseases or predict their spread around the world. On the other hand, for food quality and safety tracking, it's also very promising. Let's say we all the food chains have these systems in their hands, and then they upload this data into a common system, so that we would prevent contamination of food or other bad things about food chain. And for both systems, or for all systems, they promise very reduced costs and reduced waste. So this topic is very promising. As a conclusion, stated before times and times, deep learning and microfluidics is a very promising. They have a very promising relationship, which offers a big potential for technologies. Right now, they have some challenges to overcome, and costs are relatively high right now. But they should be developed in order to reduce this cost in the future. And once these systems are developed, they promise very reduced costs and footprints, so that we hope we will see more of this technology in the future. Thank you for listening.